Hello, my name is Diana Thompson, and I did a paper on retinitis pigmentosa. So to first understand what exactly that is, we're going to talk about what the visual cycle is and how it works. The visual cycle is the way that the body produces vision. So it starts with the light coming into the retina and touching a protein called rhodopsin. This rhodopsin is then triggered to isomerize the 11-cis retinol into all-trans retinol. The all-trans is then reduced to all-trans retinol, and then all of that goes into the retinal pigment epithelium, which then is regulated by an enzyme called RPE65, and this regulates the oxidation of the all-trans retinol into the cis retinol. From then, the rhodopsin is now active, and this activated rhodopsin binds to transducin, a GTP binding protein, and this begins a second messenger signaling pathway to produce the vision that the brain understands. So now what is retinitis pigmentosa? Retinitis pigmentosa is a progressive degradation of rod and cone photoreceptors. What does that mean? That means that the cells in the eyes around the retina die progressively throughout life. So that causes vision loss and other visual properties that affect it. So now we'll talk about what are the characteristics of RP. It's a genetic disease so it can be passed along through offspring, and ciliopathy, which means that it causes malfunction of the primary cilium that are in direct relationship with the photoreceptors, which then causes the degradation. It has rod-cone degradation, which means that the rods are degraded before the cones. It has heterogeneity, which is a characteristic that means it has a lot of variability. The same mutation in one person is not the same in somebody else, and it can have its own rates depending on person-by-person basis. It's pretty rare in that it affects 1 in 10,000 people. It's also can be syndromic or non-syndromic. Syndromic meaning that there's other symptoms that also coincide with it, or non-syndromic meaning that's the only symptom that somebody has. Some characteristics of RP are night blindness, light sensitivity, blurred vision, tunnel vision, and complete vision loss at the end or late stages. Some syndromes that are associated with RP are Bardet beetle, and this has RP, as well as post-axial polydactyly, hypogonadism, gastrointestinal abnormalities, learning disabilities, neurological dysfunction, and obesity, as well as some others. There's also Usher syndrome, which has problems, or it has vision, hearing, and vestibular impairment. There's 3 types. Usher's 1 is the most common. It has congenital deafness and prepubescent vision loss with some problems with balance. There's also USH2, which also has congenital vision loss, or hearing loss, congenital hearing loss, and vision loss in the late 30s to 40s, as well as some problems in vestibular. There's also USH3. RP symptoms occur in the 20s to 30s. Hearing loss occurs in the late teens to 30s, and partial vestibular issues occur throughout life. There are 4 ways that one can get RP through genetics. Autosomal dominant, where there's a normal gene and a mutated gene, and the mutated gene overpowers the non-mutated gene, and that shows and progresses out the disease. You get autosomal recessive, where both genes are mutated, and therefore it shows and the disease progresses. Mitochondrial, where you can this affects the mitochondrial DNA, and it can only be passed through from the mother. Get it from mom. And then X-linked, which affects the sex chromosomes of a human and other organisms. So males are typically affected more by X-linked because they only have 1 X chromosome, and if theirs is mutated, then they don't have anything to overpower it. Females can be affected too, but both X's must be affected. there are a few diagnostic tools that scientists use to determine if one person has retinitis pigmentosa. The main thing they do is sequence the DNA, and they use Sanger sequencing, next generation sequencing, polymorphism arrays, multiplex ligation probe amplifications to determine where and what gene and mutation is in their DNA. In person, an ophthalmologist can do a retinal exam, and during this retinal exam, they'll look for bone spicule pigment deposits and the reduction of the retinal vesicle. They also perform a Goldman perimeter exam, which occurs when one eye is covered and the doctor moves the light in and out of the visual field of the open eye, and then does it the same with the other eye, and this indicates where the visual field starts and stops. There's also the Humphrey field analyzer, which is one eye by one eye basis, and the eye is focused straight ahead, and then small lights are flashed at different intensities and locations and sizes, and the examinee is instructed to click a button where they see the light, and this test generates a map of the visual field based on the lights that they see. There's also an electroretinogram that can be performed, and this measures the eye's response to the light based off an electrode that is attached to the eye. So here's a normal eye that you can see. This is the ERG, electroretinogram, and here's one with somebody with retinitis pigmentosa. They have little pigment clumps, this is what they call bone spicules. And then here is a picture of a normal person's vision and how you can see everything in sight. And then retinitis pigmentosa, they lose a whole lot of their peripheral vision, causing tunnel vision. There are a few treatments that can be done for retinitis pigmentosa, although there's not a cure. So most of these just help. You can do genetic therapy, which they use a recombinant virus vector and a tyrosine mutation to stop the degradation of the capsid once it's in the body. And that can help slow the progression. You can also have oral supplements. Vitamin A has slightly been shown to work in some individuals, and some it has not. It can and is thought to slow the degradation, as well as lutein, which is a carotenoid that is found in the retina and the outer rod segments, and that has been shown that non-smokers can take lutein, and their visual field degradation is slower. You can also have stem cell transplantation, where stem cells are entered into the retina, and they can take form of the old rod and cone, and that can slow the degradation. And you can also have a prosthesis, which isn't as great because it doesn't have the greatest visual field, but if you don't have any or have an end-stage retinal disease, that can help. You can also have a pharmacological therapy, which they use a neurotrophic factor produced by neurocells to help preserve the rod and cone. And although there's not a full treatment, they are still working on it. And that's all I have for you today. Thank you for listening to my spiel, and hope you learned about retinitis.